Process of virus preservation and extraction. Vazyme Virus Sampler Stabilizer. Please read the package insert carefully before detection. This product is applicable to specimen collection from human swab specimen and preservation of viral nucleic acid. Step 1. Collecting specimen. Prepare virus sample stabilizer, tongue depressor, and throat swab. Check patient information. Clean hands. 6-step hand washing. With naturally hand drying, ask the patient to open his or her mouth. Gently press the tongue surface of the patient with a tongue depressor. For throat swab specimen, insert the swab into the deep part of pharyngeal isthmus, both sides of uvula and tonsil, and rotate the swab about 10 times against the wall of pharyngeal isthmus. Step 2. Preservation of swab. Immerse the swab head in the sample stabilizer in the tube. Carefully break and discard the swab shaft, then tighten the tube cap and put it into the disposable Ziploc bag. Step 3. Inactivation of 2019 novel coronavirus. 1. Incubate at 56 degrees for 30 minutes. 2. Balance at room temperature for 10 minutes, avoiding aerosols. 3. Gently stir and squeeze the swab. Then remove the swab. 4. Gently pipette to mix the sample stabilizer thoroughly. Step 4. Nucleic acid extraction. Treat the sample stabilizer as a specimen and operate the procedure according to the corresponding manual. For Vazyme Fast Pure Viral DNA and RNA Mini Kit, extract according to the following scheme. Aliquot 200 microliters of absolute ethanol to a new 1.5 milliliters of nuclease-free EP tube. Additional consumable. Add 500 microliters of the above sample stabilizer to the tube containing absolute ethanol, then mix thoroughly by inverting the tube. Place the adsorption column in a 2 milliliters of collection tube. Transfer the mixture to the adsorption column. Centrifuge at 12,000 grams for 1 minute. Discard the filtrate and reuse the collection tube. Add 600 microliters of washing buffer to the adsorption column. Centrifuge at 12,000 grams for 30 seconds. Discard the filtrate and reuse the collection tube. Add 600 microliters of washing buffer to the adsorption column. Centrifuge at 12,000 grams for 30 seconds. Discard the filtrate and reuse the collection tube. Centrifuge at 12,000 grams for 2 minutes. Discard the filtrate and transfer the adsorption column to a new 1.5 milliliter centrifuge tube, provided in the kit. Add 50 microliters of elution buffer. Incubate at room temperature for 1 minute. Centrifuge at 12,000 grams for 1 minute. Discard the adsorption column. The eluted DNA and RNA can be used directly in downstream experiments or stored at -20 degrees for short-term storage, or stored at -70 degrees for long-term storage.